My name is Hannah Herzog. I'm from WF West High School, and my advisor is Henry Weeks. My project is the regulation and production of alcohol dehydrogenase and D.melanogaster using gene expression, the real-time PCR machine, to determine at what percent concentration of ethanol, 0%, 5%, and 10%, the ADH gene is most prevalent. Research of the D.melanogaster not only gives us an understanding of the molecular setup of the human genome, but provides us with research on how our bodies react to different stimuli, such as alcohol. Alcohol dehydrogenase, ADH, breaks down alcohol in different organisms. ADH converts the alcohol into non-toxin byproducts so it may be excreted from the body. ADH is both in humans and D.melanogaster. Alcoholism is believed to be the cause of estimated 6 out of 100 deaths in the U.S. Testing of the effects of alcohol on D.melanogaster provides usable information to relate the human genes with the ADH proteins in fruit flies. Thus, it is possible to correlate findings from the D.melanogaster to humans and their tolerance to alcohol. More than 90% of the ethanol that is metabolized into lipid in larvae goes through a pathway that is dependent on the ADH activity. J. A. McKenzie tested the tolerance of D.melanogaster with different alcohol percent concentrations at 0%, 6%, and 9%. McKenzie found no outward difference in the D.melanogaster's behavior at these different levels. Since then, different researchers have found an alcohol tolerance medium between 0.5% and 13%. In this experiment, we will be testing alcohol concentrations of 0%, 1%, and 5%, and 10%. The purpose of this experiment was to test the regulation and production of alcohol dehydrogenase in D.melanogaster using gene expression, real-time PCR, to determine at what percent concentration of ethanol the gene is most prevalent. It is predicted that ADH will be more active the higher percentage of alcohol is present in the fly's environment. This is because the flies need to break down more alcohol more quickly. In this experiment, we will have 7 D.melanogaster cultures set up with varying degrees of alcohol. 1 at 0, 2 at 1%, 2 at 5%, and 2 at 10%. RNA will then be extracted after 24 hours and converted to cDNA. cDNA is then run through real-time PCR machine. The real-time PCR machine is used to amplify and simultaneously quantify a targeted DNA molecule. In this case, it is the ADH that it is targeting. The amount of an expressed gene in a cell can be measured by the number of copies of mRNA transcript of that gene present in the sample. In order to robustly detect and quantify gene expressions from small amounts of RNA, amplification of the gene transcript is necessary. In this experiment, the custom ADH assay will be used to locate the 771 base pair ADH gene so it may be detected and quantified. Phase 1. Preparing fly cultures. We will have one control of 0%, 2 of 1%, 2 of 5%, and 2 of 10%. The alcohol used is a 40% vodka alcohol. Using the C1V1 calculations, we can find the percent of alcohol at 1%, 5%, and 10% to have a pure alcohol concentration. Phase 2. RNA extraction. After 24 hours of the flies being exposed to the various environments of alcohol, we will use the fly tissue will be distributed by a pestle, and we will use the RNaqueous 4 PCR kit by Life Technologies to make a total of 7 reactions. In phase 3, RNA to cDNA. The Tachman reverse transcription reagents kit from Life Technologies will be used to convert our RNA to our cDNA. Our nanodrop was used to measure an RNA concentration. The samples were then placed into the PCR machine. Phase 4. Real-time PCR to measure gene expression levels. The concentration of the RNA was found by using the nanodrop. The real-time PCR machine was then set to run under one cycle of 95 degrees Celsius for 10 minutes, then 40 cycles of 95 degrees Celsius for 15 seconds, 60 degrees Celsius for 1 minute, and finally infinitely hold at 4 degrees Celsius. My results. Our real-time PCR was loaded in our 1% A and our 1% B, our 5% A and our 5% B, our 10% A and our 10% B group. We had one control at 0% and one 18S control to make sure that the cDNA was transferred correctly. Our amplification plot shows our 1% here, the dark red, and our 5% at group B, the blue right here. Then our 5% A, the green yellow here. Then our 5% B, the green yellow here. Our 10% here, A, and here, B. Then we have our 0% control here, and then we have our 18S control here. On our plot, it shows that no amplification occurred, therefore no conclusive results can be concluded. The ADH gene has no amplification on the real-time PCR machine, but the 18S control did. This shows that my conversion to cDNA was successful. It is possible that the assay used did not express the correct area of the protein. What is known is that an error occurred during the phase 4 gene expression using real-time PCR. Due to the small amount of flag colonies used, there was a low concentration of RNA to work with. This may have had an effect on my inconclusive results. Alcohol is in both humans and D.melanogaster. ADH is a gene that breaks down this alcohol so it can be transported out of the body. To understand ADH in ourselves, the gene is studied much more frequently on D.melanogaster. The purpose of this experiment was to test ADH gene and their production in D.melanogaster using gene expression to determine at what % concentration of ethanol the gene is most prevalent. To quantify the ADH level, RNA was extracted and converted into cDNA, and that was run on the real-time PCR machine. The results of the real-time PCR machine were analyzed to see whether the ADH gene became most prevalent. The ADH gene had no amplification, however. More research will be necessary to figure out why no amplification occurred.